I'm Joe Loscalzo, and I'm moderating this panel discussion on genomics and precision drug therapy. Joining me are Mary Relling, George Patrinos, Charles Rotimi, Peter Turnbaugh, Amalio Telenti, Gert-Jan van Ommen, and Razelle Kurzrock. And the way we're organizing this session is that I'll just give an introductory slide, and then each of the panelists will, beginning with Mary at the other end, provide a couple of slides of insight and stimulating and provocative comments that we'll use to generate more discussion at the end. We have some general questions for discussion at the end, and we'll also obviously open up to the audience your input for relevant questions or discussion. So if I could have slide on. Okay. It's on my laptop, but not on the screen. No, not yet.
I got it. Sorry.
Oh, great. Sorry.
I can use this if you'd like.
Okay, thank you. So just to set the stage, I illustrate for you here a couple of points I'd like to make about this notion of genomics and precision drug therapy. This slide is taken from a review published a couple of years ago with my colleague Jane Leopold. On the left, we see a group of individuals with disease A who have a common, inclusive, reductionist endophenotype. They've been selectively genotyped as a population, and therapy is initiated based on the genotyping for that phenotype. But we now know, of course, that in order to ensure that precision therapy is truly precise, one needs to develop greater strategies for deeper phenotyping. Inclusion is not the word, but subtyping is. This common endophenotype is then segregated based on a host of genomic strata into subcategories. And these subcategories can be refined and defined on the basis of a variety of types of network analyses, like what I like to call now reticulomics, from the Latin reticulum meaning network, where one generates a drug target network that is based on, for example, the protein-protein interaction network for the proteins that are responsible for the pathophenotype of interest. And variants in particular drug targets within each of those individualized reticulomes may be then used to guide precise therapies for the subphenotypes for this disease A. And if we keep that in mind, the disease could be your favorite malignancy, it can be hypertension, all the diseases with which we're quite familiar as having been increasingly inclusive for purposes of easy understanding in conventional commercial drug development, I think need to be soundly eliminated, and the field needs to move in the direction of appropriate and economically feasible subphenotyping for appropriate targeted therapy. So I will next then ask Mary Relling from St. Jude's to talk about her, or provide her comments about this topic next. Here, do you want to?
Sure.
Actually, if you'd like, just pass it down.
Oh, okay.
Or stand, whatever you want.
No, that's good. Thank you. And it's interesting in just a hallway conversation before this started, you know, let's remember that the vast majority of pharmacogenomics is never going to make its way into the clinic as being actionable at all, certainly not at a single gene level. And I'm sure in my other life, I do research without bounds, looking at the genomic determinants of variability in response in children with leukemia. But in my life as leader of CPIC, I am trying to focus on those few genes that have such penetrant effects in essentially a monogenic fashion on the variability in drug response that they are ready for actionability based on genomic variation that we can measure. So I showed this slide yesterday to make the point that even for those very, very few drugs that have made it to the point where an FDA drug label either recommends or requires that a genetic test be used prior to prescribing, only 1.5% of prescriptions at a major academic medical center in the US were accompanied by genetic testing. So you 600 people are fantastic, and you are leaders, and you're going to be propelling this field forward, but we need funders and regulators and the public to understand that the use of pharmacogenomics in the clinic is still incredibly, incredibly rare, as much as we're loving to hear about it all today. And that I do think that we still, unfortunately, are in this position where a big reason that people aren't doing this in the clinic is that it's hard to do. And getting a genomic result off of either an array or a gene-specific test or next-gen sequencing that says at position 1632,478, there's a C to a T, translating that into don't use mercaptopurine in this patient or use a 10-fold dose reduction in this patient is a process that requires an incredible amount of number of different steps. It's requiring an incredible manual curation of scientific literature and understanding of both the drugs involved, the genes involved, and also the diseases and adverse effects. And one of the, I guess, other take-home points that I think we have to figure out how to grapple with is that this is not a static problem. So if somebody, gave us a huge amount of money and we could write every single CPIC guideline because we had 400 fantastically talented people to go through the literature in the next year and summarize where we are and get every CPIC guideline done by the end of the year, we'd have a pile of work waiting for us because data continually evolve. And I don't see that the data evolution is going to slow down. We need to figure out as a world how to fund efforts to continue to curate these gene-drug relationships in the case of pharmacogenetics so that we can continually update our practices to provide the best possible prescribing for patients. And unfortunately, that's not very sexy. That's not something that companies like to fund. That's not something that NIH likes to fund. That's not something that my institution likes to fund. But there's only a few really talented people who have the wherewithal and knowledge of how to do this. And I don't know how we're going to make this extensible over time, but we need to figure it out if we want precision medicine to not only be implemented, but be ongoing. I'll turn it over to George.
Thank you. George?
So thanks so much. It's great to be here. Come from the other side of the planet, basically. And it's really nice to see all this, room full with so many people interested in pharmacogenomics. So this slide basically is to indicate that I also, have a regulatory role. So I'll try to share also my experience from this part. Joe asked me he asked us basically as our moderator to summarize in 2 slides what's more appealing for us. So I'll try to put some points here to spark the discussion. In 2014, when we published the commentary NEMBO reports, the designer depicted this as the future before and after pharmacogenomics. So it was quite intriguing to see that pharmacogenomics, actually the life before pharmacogenomics looked like a black box. So once you start revealing your genomic differences that could be delineated with a response to certain drug, then the box starts to take some colors. Too many gray boxes, of course, but we can hope that in the future this will also take some color. How this can be done? First of all, we need to generate evidence. We heard yesterday Gert-Jan describing the PREPARE study. There have been some other studies that have been done in the past, like the PREDICT trial in Vanderbilt. So we need to have evidence to generate to demonstrate that pharmacogenomics is important and a key element for future medicine. Secondly, we would need to try to harmonize the existing drug dosing guidelines. We heard Mary yesterday and today speaking about the guidelines have been generated by CPIC. Unfortunately, we have seen that there have been some big discrepancies in the guidelines that have been produced by FDA and the EMA. So this needs to be harmonized, and the differences are big, trust me. One important element is to bridge the educational gap about pharmacogenomics. We asked actually to a large number of healthcare professionals and we asked them whether adenine pairs with cytosine, and 19% said yes. So we need to address the educational gap on genomics, but also on pharmacogenomics, so that we can help the physician understand what the researcher says, what the laboratory science says. And this can be done in two ways. Firstly, we need to design tools. We need to design some tools, IT tools, to try to translate the information as it comes from the lab. Secondly, we need to ensure that we can have educational pharmacogenomics. This can be also, based on a harmonized manner, because we have seen that there are big differences in the educational courses on pharmacogenomics between Northwest European countries and Southeastern European countries. And the same is actually now true for Latin American countries, for Middle East, and also Southeast Asia. This is not just the US and Britain, that everything is very harmonized, very high profile. These countries should also address this particular need. And lastly, an important issue is pricing and reimbursement. We may generate a lot of evidence, and we may have all the best tests in the world. If we don't show that this is affordable and we don't show that this is cost effective, then this is all for nothing, right? And for this reason, there's also this PARC consortium that has been formed recently, the pricing and reimbursement, which try to address this need. So what are the opportunities? In my view, we have 6 opportunities here, 6 points. First of all is not just pharmacogenomics, we're talking about population pharmacogenomics. To this end, there have been 2 major projects that have been, they were running in the past. First is the EuroPGX project to try to demonstrate the allelic spectrum of pharmacogenomic biomarkers in several European countries, and now the same is being replicated for Southeast Asia for 100 pharmacogene projects, using a panel of NGS-based panel for 100 pharmacogenes. Secondly is to develop a tool. To try to get the result from the lab. If it can be few variants, or it could be an NGS-based panel, and then try to translate this and link it to the existing guidelines, so that the physician doesn't need to search through the literature or through the websites, but he could basically identify this through with the use of the tool, so that he could prescribe the right dose to the right patient. We need to establish educational modules on pharmacogenomics. This could be e-learning modules or blended learning, combined electronic learning together with on-site training. We should put emphasis on health technology assessment for drug-guided interventions, so that we could assist the policymakers for pharmacogenomic testing reimbursement. We should try to use this information for a new type of clinical trials, which is the genome stratified clinical trials. And last but not least, we have heard all these beautiful interventions, especially this morning we heard Peter and Amalio discussing about microbiomics, about pharmacometabolomics. It's great. If it costs €2,000 or $2,000 or £2,000, this would not be applicable to countries like Ghana, it's like Thailand, Malaysia, Chile, and so on. So we should think globally. We should not just think, on a country-specific basis. So how we could harness, we should find a way to harness the potential of developing countries. You see there a book that has been produced recently with Catalina, Correa Lopez, and myself about demonstrating the potential that genomic medicine may have in developing countries. So imagine a restaurant. If you go to a restaurant, you don't order the full menu, right? You select dishes. So you start with studies that are performed in developed nations, like the US, the UK. You find relevant biomarkers for genome-guided intervention, and then you select which of those biomarkers could be relevant to specific countries. So, in other words, you don't take the full menu, you just identify what is relevant for certain countries. And this is, in fact, what we recently called as the second fast winner strategy for precision medicine. So, over to you, Charles.
Okay. Again, glad to be here. I just wanted to share with you some of the way I think about genomics in a global context. One of my biggest worries is that as we move fast in developed countries, that we are indeed maybe compromising what we can learn by not engaging the larger global population. So basically, the question I like to ask is, will tomorrow's medicine and technology work for all human population? And I think the only way we can answer that is to truly engage global populations in a way that we can, systematically document human genetic variation. One of the things that ways I try to express this when I go around the world to give talks is that this era of precision medicine, what does it really mean? To me, what it means is that I cannot rely on my brother's measurement for my suit or my pants. Otherwise, it will not fit. That's how close we have to be in terms of individual understanding of human genetic variation. So really, all of us, whatever the community we are in, whatever reasons we may or may not want to participate in genomics, we need to reconsider that, because as we do pharmacogenomics, it's really you. Even your mother sometimes cannot represent you because she may not carry that variant that you have, or may react to it very differently. And I'll give you an example. This will tell you how old I am. I grew up in Nigeria some years ago. I usually say 18 plus X, something like that. But really, I used to get malaria like twice a year. And then the treatment that we take is chloroquine. And my mother takes chloroquine, I take chloroquine. It helps us with our malaria, but she's fine, but I itch for days. And so if you're interested in the pharmacogenetics of response to chloroquine, myself and my mother, we're going to belong to different groups. So that's how precise we have to be. So we really need to engage, our global populations. And the other thing is that by not engaging global populations, we are actually shortchanging our understanding of variation that are relevant for drug metabolism. And one of the studies that we published some years ago really demonstrated this, even in terms of warfarin dosing, that there are haplotypes that exist in African populations that are actually quite common, that may be relevant to warfarin dosing that is currently not being considered. So again, that to me is really important in terms of engaging global populations. We've also seen recently that some of the things we are calling pathogenic in European population may not necessarily be pathogenic in African populations, and they might indeed be quite common. So again, it will help us to understand biology and physiology if we engage global populations. The other point I want to make is the way we describe pharmacogenomics variation, we need to be extremely careful, even human genetic variation in general, because sometimes when we find something that is more common in one part of the world, we tend to label it with that part of the world, which can distort our understanding or even public health decisions that needs to be made. And the example that myself and Lynn George gave in this paper we wrote some years ago is really the hyperreactivity to this HLA variant in terms of negative predictive value for reaction to avocado. So when you look at the Masai population, for example, just using the HapMap data, we see that Maasai carry probably one of the highest frequency of these variants, whereas if you look at among the Yorubas, which is just maybe 500 miles away, the frequency is almost zero, and African Americans about 1%. So when you use labels like Black or African, and this, you distort our understanding of this, and that indeed can lead to how we evaluate public health strategy. So my concern is that as we find these things and understand variation, we need to be careful, even the papers that we write, how we label this variation. Thank you. And I move to Peter.
Okay. Well, I'll try to be quick so we have time for the discussion, but I mean, I think all of the points that have come up earlier, I would just agree with and, replace human genetics with the microbiome, or maybe add the microbiome to human genetics. But I just wanted to sort of talk about a couple ways that we think about the different layers of the microbiome, and so maybe later we can discuss how to tackle each of them. So we've sort of talked about a lot of this morning. There's complex communities that are in the gut and in other sites in and on the body. They're composed of these individual members, and so you can either sort of think about them at the community level or at the species or strain level, genes, enzymes, and metabolites. And in terms of sort of diagnostics, or like what sorts of things we might want to measure, you can measure at any of those levels, and so we can argue about which one's better or worse. And then, in terms of targeting, there's really no technique right now to target genes. You could potentially change metabolites somehow, but I think most of what we talk about in terms of manipulating the microbiome is in terms of targeting specific members at the cell level or at the enzyme level. And then on the metabolite level, we often think about this as, finding new drugs from microbial communities. And so microbial metabolites that we might want to use as the next generation of therapeutics. Okay. And so, this is sort of a diagram that's just our microbiome version of what we already saw. The sort of classical approach to medicine where you just, do the trial and error until everybody's treated. Or you give up. And then, pharmacogenomics has this incredible excitement still around, the idea of using the human genome to better predict ahead of time. And so, what we are working on and very interested in the microbiome field is how you now add pieces of microbiome research to these personalized treatment plans. And so these are just a few sort of ideas for discussion. I think, one of the things we struggle with is that there's thousands of drugs on the market, and so, which ones are actually the most important, which ones are the hardest to predict right now, either by trial and error or by pharmacogenomics, and which ones, do we think could feasibly be affected by the microbiome. Can we do this in a predictive way? Is it, do we have to study every drug in the lab before we can, recommend something, or are we going to get to the point where we can actually predict? And also, what sort of either preclinical or clinical data would you guys want to see before you're convinced that you actually want to incorporate the microbiome into these clinical studies? So, turn it over for me.
Thank you. So I want to bring two ideas to this table. First is the issue of clinical trials for pharmacogenetics, and I say clearly not clinical trials with pharmacogenetics, it's clinical trials device for pharmacogenetics. The successful one was PREDICT that made the label for abacavir. But, and you probably know more than me, but it's difficult to find tons of clinical trial or attempts, which is probably the main limitation of how the uptake of pharmacogenetics has taken place in the clinics. And there's a second concept, is that many clinical trials concepts are based on either a single drug or some severe drug effect, which is of course very important. But there's something more, it is the general issue of tolerability. And on the right panel you see this plot, is a Kaplan-Meier of just patient discontinuation of a drug because of intolerance. And we can detect that. Well, is this serious for the patient? No, the patient just comes back and says, this drug makes me, I cannot sleep, or makes me dizzy. And we change the drug and everybody feels good about it. Well, that's more medical visits, there's more cost. Now, you could probably make a clinical trial just of general outcomes. Does it make sense on medical health outcomes? Does it make on visits? Does it pay off, etc.? So I think we should not be so dogmatic about the severity or the importance of the drug effect, and more general saying, well, does pharmacogenetic reduce or contribute to medical care in general? And I think this example that was for the medication efavirenz was a very good one. There's a huge difference between having a slow metabolizer, that's what we are detecting for efavirenz, or not. Is it important? Well, not really. The same thing was done also for atasanavir and Gilbert syndrome. Does the patient get sick? No, it just gets yellow. What happens? They ask for a change in drug, and that costs. So I think we should have more clinical trials if we wish to have the payers pay for it and the clinicians using it. And the second concept, maybe something more unfamiliar to you and more familiar in the genomics arena, is we always look at genomics as an active ingredient. We query, and I think I've heard many times today people saying, well, how can we interpret? Well, there's something probably much more interesting, that is the passive use of genomic information. Think of it about Google Maps. You are not querying Google Maps. The Google Maps knows where you are and it's going to tell you what's the restaurant you're looking for, the gas station. So for me, pharmacogenomics will be a prime example of passive use of the genome. Your genome on the phone will know when you are in the pharmacy. You can beam towards the code bar of the drug, and it will tell the pharmacist or the doctor there may be an issue with this prescription. You are not querying, particularly not as a lay person, you are not querying the genome. You are actually, the genome is talking to you when and if needed. This also will avoid these reports that confuse everybody, where we go all over the place about this cytochrome and this allele and this C2T and T2G that drives people nuts, because that's not what they want to hear. What they want to hear is there is an issue with this prescription. And I think, I hope you give a thought to this and place the genome in a way that probably will be more easily processable.
Well, thank you very much. My name is Gert-Jan van Ommen from Leiden University. Let's put this here. In the time that the genome actually was sort of fished out of the haze, in 2000, I was president of UGO, and there was a lot of commotion about it in a way, and so I'd like to bring this to the attention. But other than the commotion about the fact was there was a race between the public project and Craig Venter, and it was found by the newspapers that the public was the winner. And the way they meant it at the time was true, that then many laboratories worldwide suddenly had access to something that otherwise would be precluded to some well-endowed laboratories in the US. But in fact, in another sense also, the expectation was raised to the right way on what we're talking about. And what we're now here for is the sort of personalized precision aspect of it. And it actually has three aspects: it's diagnostics, therapies, and prevention. And in the diagnostics field, at the time when I was president of UGO, and many other people with me were accosted quite often by the rare disease patient community saying, oh, you're just sort of burning up a fortune and creating a big pink elephant which will fall asleep in the corner of the room, and it's of no use to us. Well, two things on that. One is, if the Americans have left Iraq 4 days earlier, they would have paid like 10 times the Human Genome Project all over. So it's relative what you invest in here. Second, actually, the rare disease community is the only community so far that I know that really has benefited from the genome, with a tremendous amount of personalized diagnostics for these rare diseases. Now, for the therapies, we're now in the field. It's the cancer field which is benefited and is benefited from it, and the pharmacogenetics field is benefiting from it. But it still is early days, because we still don't go into the rare variants that jointly have a very strong predictive effect. People have discovered that, so they went into biobanking quite seriously. Biobanking was recognized as one of the main things to do. And within 5 years after 2000, from 2007 onwards to 2012, there was a phenomenal amount of GWAS studies done, as you can see on the screen, for many mechanisms that was extremely useful. They also try to apply that in, say, predictive, what I discussed with my neighbor, genomic risk scores, and they were originally not very useful. And the reason for that was that they went for the common variants, which is, like, say, the star allele type of approach in pharmacogenomics. Go for the common things where you can do the predictions. But nowadays, and this paper is really recent, Nature Genetics in September, there are the first genome-wide polygenic scores for common diseases that actually identify individuals with risks equivalent to monogenic mutation. So why would you screen for monogenic mutations and not take along these? Well, first of all, then you're not talking about a risk score with a panel of, like, say, 10 genes. You're talking a risk score of a panel from, say, 4,000 to 7 million items on the map. So it's really a different game. And what you discover is not mechanistic at all. So it doesn't give any clue directly for how to treat diseases and understand the mechanisms, because you still look at an enormous cloud of mechanisms. But still, this approach identifies 8, 6, 3, 3, and 1.5% of the population at greater than threefold increased risk for several major diseases: coronary artery, atrial fibrillation, type 2 diabetes, as one rule followed away, inflammatory bowel disease, and breast cancer. So there's something coming on in that field which uses the genome precisely as my neighbor said, as a static thing that you could sort of use when it has been identified. And there is the way of identifying this is low-hanging fruit. One of the posters and people present here from Leiden University is poster 638 and from Friday, and it'll go in some detail on this because it actually, I'll go back. It tries to repurpose whole exome sequencing that's been abundantly available nowadays for personalized medicine. And you can actually use that. One of the main findings is that all the possible pharmacogenomics phenotype calls, about 3/4 of them can actually be extracted from whole exome sequencing. That's done nowadays. So that's worth a try. And the other things, I'll leave you to go to the poster, but the last one is also quite important. Nowadays, it's hard without having father, mother, and child to find out what the phase is of the variants. And this is important in pharmacogenomics as well, because sometimes when they're on the same allele, you have a different reaction for drugs than if they're on two different alleles. And that's also something what you can do if you have a trio-based genetic information. So I think that this type of precision really needs the two fields to merge much more than they do nowadays. They are there, they're big, they wave at each other, and they actually have sort of complementary meetings like this one. But at some point, they should really get real wet and join the fray. And I think that this is one sort of step from the star alleles to use whole exome and whole genome as a basis for a static multivariant prediction of any drug that people might be needing in their life. And now over to my colleague Razelle Kurzrock.
Well, thank you. Actually, I've really enjoyed hearing the wonderful comments and presentations. It's been a real learning for me as well. So I want to emphasize what came at the end of my talk, and because I think, to me, this is a really fascinating area, and that's the immune pharmacogenome. I showed this slide before, but I want to go through it because I think it's really important. In cancer, we have really concentrated on genomics and much less so on the host. And yet the host is responsible for many aspects, for toxicity, certainly, and in maybe for response, and certainly for immune presentation. Immunotherapy is one of the areas that everybody is most excited about. One of the parts of precision medicine that has really begun to transform cancer, but we're just beginning to understand how immunotherapy works. This, to me, is absolutely fascinating. So what we understand about some of our new immunotherapies, like the checkpoint inhibitor drugs, that are now, the Nobel Prize was just awarded a few days ago for discovering these drugs. Why was the Nobel Prize awarded? Because about 15% of patients respond, and some of those responses are really quite something. We've never seen this before. Patients that are end stage, maybe ready to go to hospice, and then they're in complete remission. They're still in complete remission 2 or 3 years later. How does this happen? The immune system has figured out how, the tumor has figured out how to deactivate the immune system. It exists because it has turned off the immune system. What the checkpoint inhibitors do is that they allow the immune system to be reactivated. But once reactivated, there's still another step, and it's really important. The immune system actually has to recognize the tumor. It has to differentiate the tumor from normal cells. And one of the most important ways it does that are by the neoantigens that are presented. The neoantigens are presented by cells, and they're a product of the mutations. And so patients that have very few mutations and therefore very few neoantigens have a very small chance that the immune system will recognize this as a tumor cell that needs eradication. But patients that have a lot more mutations, it's throwing the dice. You have a lot bigger chance that you have an immunogenic mutation. But it's not just about the good luck of having enough mutations that the immune system can recognize. It's about your endogenous MHC, which presents those mutations. And it's really, I'm still amazed by the structure of the MHC. It's like holding up a sign with the arms to the T cells and saying, here is this mutation I'm carrying. Do I belong here? It's known that different MHCs from different individuals present different neoantigens in their own way. And we can now calculate how well a particular individual's MHC will present those neoantigens. And that can help us. We've already seen this in the clinic. I've showed a little bit of our preliminary data. That can help us predict which patients will respond to immunotherapy. So it's not just about the mutations, the somatic mutations. It's about the host and how well they present those somatic mutations to the immune system once our drugs have reactivated the immune system. Thank you.
Well, thank you. I'll leave this slide up as sort of background as we open the panel session up for discussion. And let me begin with a couple of questions that derive from the comments that you've all made. And the first is, it sounds, I think all of us would agree that there's a need to consider some subcharacterization of subpopulations for precision medicine to be successful in pharmacogenomics as well as in other areas. And my question is, assuming you all agree with that point, how should we begin to subcharacterize these subpopulations? Should it be ethnicity-based? Should it be microbiome-based? Should it be deep phenotyping-based with respect to a particular element of phenotype that's relevant to a therapy? Should we develop an index of some sort that incorporates these and other elements that can be quantitatively parameterized? What are your thoughts about that?
How about genomically based?
Well, that's where we began, but that's part of the problem.
Well, but that's easy. And then that takes care of a lot of the ancestry-related.
That's the ethnicity side.
Yes. As we are reading about with Elizabeth Warren's 23andMe, works better in some cases than others. But I guess I think that's a great starting place because it's invariant, because it doesn't change as the patient ages, because it's, at least let's pretend that it's tissue agnostic and not worry too much about mosaicism. Of course, as a clinical pharmacist, I mean, the other things that really affect drug efficacy are things like adherence to therapy, which is a major problem, which most of us ignore, age, renal function, liver function, drug interactions. I guess I have been shocked, since I've been doing this now for 35 years, that we still don't really have good tools in the clinic to help us integrate all of that very basic information about the individual that would help precision medicine get off the ground right away, even if you didn't have genomics. We still don't have very useful systems in our clinic for guiding prescribers with what to do with those basic things: drug interactions, renal function, liver function, diet, age. Incorporating genomics is not trivial, but it can be done, and I think that if we just did those things, we would go a long way to improving precision medicine in the clinic without any more research, without any more tools, without just, if we just had better implementation systems.
Okay. Actually, I think Gert-Jan was going to make a comment. Please.
Yeah, one thing, I would second the genome first type of approach. But you can perhaps combine it, making it a bit easier for yourself when you would do a genome first and drug first approach rather than a phenotype, deep phenotyping. And the trouble with the pharmacogenomics, setting it apart from, say, rare diseases and cancer and so on, is where's the phenotype? The phenotype's only there when you do a treatment. So it's a bit like any gene environment thing. You have a loaded gun or an unloaded gun, and a gun with a trigger or no trigger. If you have an unloaded gun, you can pull the trigger whatever you want, nothing happens. If you have a loaded gun without a trigger, there's no problem. And in many cases, people are loaded guns with no triggers. And so having to pull the trigger all the time for presenting the phenotype with the drugs is difficult. So I think going for the genome first, but then the ethnicity, what you implied, I would say it population, because many populations themselves are blends. The Americas, even the white population, is a blend from many places in Europe. And what you probably would need is have some sort of basic reference population, which is not different for any other type 2 diabetes or whatever approach. And Europe and America should put power behind taking those generating those reference genomes of big enough, because using a genomic risk score of the wrong population is not going to help you a lot. But if you help them, and if you actually do have them, then you can do a sort of basic panel-based slotting of an individual where they are on this sort of reference map, and then from there go to what type of drug reactions you might actually expect if you look for certain drugs rather than on, and then prioritize the drugs for which you do this.
Okay. Yes, George.
Yeah, so I agree with Gert-Jan and Mary. I think genomics should be the foundation. However, we need to adopt a more holistic approach because genomics could be easily done. The moment you're born, you can have your genomic information that's going to stay stable until the end of your life. But other approaches like metabolomics, for example, could give us a certain phenotypic snapshot of a certain situation. So if a test to be widely applicable needs to be quick, cheap, and relatively easy to perform. So I think genomics should be the basis, but other approaches could be also used to make it, in fact, to maximize the utility of pharmacogenomics.
Okay, thank you. Amalio?
I just want to make a point on data representation. I think one of the things we need to confront is why in medicine we are not as good as other sectors of society on representing people. So if you ask, again, Amazon or Google or Netflix, they know how to represent people. They know how to they create a vector of yourself. So I think there's a lot of science, there's a lot of data science available that we probably should embrace, and we're probably pretty shy about doing so. So independent of whether we put the genomics at the base or the EHR at the basis or whatever other technology, we will not go very far away if we don't identify the appropriate tools for human representation. And once that's improved, I think everything like finding anybody who looks like me and therefore will probably have the same outcome or the same effect will become a tractable problem. I think healthcare needs to look into how other sectors have solved data management and representation.
Thank you. Razelle, did you have a comment?
Yeah, I'm not sure I understood the question, but in the field of cancer, I think genomics is the basis, but it's certainly not the end. Clearly, things that are seen at the genomic level may be silenced or not expressed at the transcriptomic or the proteomic level. There's metabolomics, which may give you another snapshot. As far as to whom this is applicable, there's actually a lot of controversy about this. I'm not sure why, but I actually think it's applicable to all patients with cancer, and that we're standing at the threshold of real change where it will be applied. As the prior speaker said, it's the implementation. We're very behind in the implementation. But I can't think of, even in the curable cancers, where we couldn't do better. We have curable cancers, but often they're cured with therapies that leave patients quite scarred in many ways. We could do better with the therapies. We could decrease the toxicity. We could figure out who doesn't need adjuvant chemotherapy because we give a lot of patients toxic drugs that don't need them. So I don't even think in even the best cancers where we're curing large fractions, even in those cancers we could do better. And then in metastatic solid tumors, which still remain a death sentence for most people, this is the key to really transforming that landscape in the next few years.
Well, I'm glad you all sort of played into my next question by emphasizing genomics as a first approach, because as I reflected on the issue about why it's so difficult for pharmacogenomics and genomics in general, clinical genomics, to be taken up by the clinical community, the major reason is that we give mixed messages. What's a variant of uncertain significance? What are the implications of having 6 variants, only 2 of which have been linked in some subgroup to a phenotype X? A fine example of this, of course, is hypertrophic cardiomyopathy. 250 odd mutations that have been variably linked to phenotypes which may or may not be anything more than an echocardiographic difference in the wall thickness of the left ventricle, and may or may not have consequences, real clinical consequences for heart failure or sudden death. Man Rai wrote a really nice paper in the New England Journal with Zach Kohani looking at ethnicity differences and the absolute lack of relevance of so-called variants of a known effect in those subgroups. So until we get our act together in screening sufficiently broad numbers of patients with whom we have real phenotypes, as one of you mentioned, especially with respect to pharmacogenomics, that we can characterize as robust outcomes, I think we're always going to be in this difficult situation of trying to convey the education that's needed for the practitioner. And I would raise one other point that I think is interesting, having to do with this continued reliance on the one-off associations. A couple of you in your presentations earlier made the case, and I think, Amalio, you said it nicely, that thinking about these relationships of a genotype to a phenotype in a conventional reductionist strategy is frankly worthless, except for extreme Mendelian disorders that we're not dealing with very much here in complex adult disease. And therefore, we really need to think about gene by gene and gene by environment interactions in a more robust, quantifiable, structural way. One way, of course, an area in which I work is to create networks of interactions among proteins whose variation in sequence affects the strengths of association and as a consequence affect pathways that are relevant to the phenotype in the network. And I'll give you one example and then open this up to the audience. My colleague, Jörg Menke, in our paper published a couple of years ago in which we looked at the modules in the protein-protein interactome that reflect subnetworks governing 265 human diseases, found that in the case of the diabetes, type 2 diabetes module, if you look at the 215 or so loci that had been linked to type 2 diabetes statistically significantly by GWAS, as you know, about 200 of them have a very tiny effect size. But if you map those loci, those genes, to the interactome and monitor the z-score as a function of mapping one by each, you begin to see clusters form that would not be appreciated, is not appreciable, is not able to be appreciated by conventional GWAS or any other association. And these clusters then begin to create pathways that had not previously been recognized. So now you can have a more reflective view of the genetic and genomic context within which a variant might be operating that can govern a molecular phenotype that might be actionable. So I think it's not the only way to approach the problem, but I think there are strategies of this sort that the folks, the mathematicians in the complexity field and physics and applied math fields have been working on for years for the kinds of systems in which they work that are readily applicable at this point in time to the kinds of biological questions we're all interested in, including those dealing with the effects of drugs. But let me open it up to the audience. We have about 40 minutes for questions and discussion that you can bring to us. Yes, I think you got up first.
Hi, my name is Adriana Sperlia. I'm from the PhD in Bioinformatics program at UCLA. So this question is for Mary Relling. You mentioned how time-consuming the kind of work that CPIC does is. So I was wondering do you see value in potentially automating these things? Do you see promise in maybe some of these NLP-type approaches to these things, or whether it's either that or maybe pushing publications to summarize things in a way that is machine-readable, or just kind of what are your thoughts on that?
So I see Michelle Carrillo at the back of the room, my colleague at PharmGKB, where they have used NLP and other tools to help summarize some literature, gene-drug associations, and made a lot of progress there, and that information is used by our guideline authors and our curators when we start summarizing the evidence that underlies every CPIC gene-drug pair guideline. But I guess it's remarkable the extent to which it still takes a human reading every single article to come up with these very nuanced clinical interpretations of how would you actually act on that. And to my knowledge, there is no way to automate that. Many people, a lot smarter than I am, have been trying to do that for many years, and it still comes down to clinically savvy scientists and clinicians reading the literature to weigh the associations between gene variants, side effects, desired effects, role of alternative therapy. There's no way to automate all of that process, unfortunately. And so that's why I'm so concerned that this is going to be a process that's going to take a lot of manpower with no end in sight. And so is our society really willing to devote the resources that are going to be needed to continually evaluate how the medical literature feeds into this decision making?
Thank you.
Yes.
Yeah, hi, my name's Steve Shearer. I'm at the Genome Center at Baylor College of Medicine. We've recently completed the sequencing on 10,000 individuals from the Mayo Clinic, wherein we applied the third generation of the PGRN-seq panel to these individuals in a prospective fashion to be incorporated into the electronic medical record. And I'll bring up 2 points for the panel. One of the challenges that we had was basically identifying enough truth set samples to get CAP-CLIA certification on this panel and the need for those kinds of resources. And secondly, the need for institutional advocates to really push this. Without Dick Winchelbaum at the Mayo Clinic, this never would have happened. And there's a certain inertia, if you will, in the clinical field to really needing or thinking they want to adopt this. And I'd like to hear the thoughts of the panel with regards to all this.
Who would like to comment? Gert-Jan, would you like to?
Well, I think one of the second one hits the nail right on the head. You're not the only place where there is not a lot of enthusiasm for the actual implementation. I think that our problem with this field is much less technical or even scientific, but much more social. And it has to do with, as long as there are mixed signals from the reimbursement authorities on whether or not you should or you should not use this, it means that you'll never have somebody that takes a decision that ultimately 10 years from now puts him or her in the same seat as big tobacco. And for that reason, people just sort of try to duck the bullet because it's just damn complicated to integrate all these mixed signals at present. And I just don't have a full-blown answer, but at the same time, if 95% of the people at least have one actionable gene that can be linked to a drug that's being prescribed to them in the next 3 years, that's no longer like 5%, that's 19 out of 20. And that means that somehow in the near future, I will see an increase in the responsibility for people not taking those decisions on board, simply for that reason. I mean, it's crazy if you compare the amount of money that still is being poured into high energy physics and astronomy. While we know more or less what happens in the first minute after the Big Bang, and we know more or less what we look at when we look between two stars, and we can't even predict the effect of regular drugs within 50% precision. That's nuts.
George?
Yeah, so I agree with Gert-Jan that that's also social, because also the people should be aware of the value of genomics information, but it's also political, because if you are to make this part of the healthcare system to get reimbursed, then the politician will ask you how much money will it cost, and we need to demonstrate that the new intervention doesn't necessarily need to be cheaper. It will be more expensive perhaps because it's a new intervention, but we should demonstrate that it's going to be cost effective. So how much will contribute to improve the quality of life of the patients? So this is why the reimbursement and all these efforts to demonstrate the cost effectiveness of the new genome guide intervention are quite important. So it should go, we need this information to be able to demonstrate to the politicians that it's really something worth doing.
So,
Sorry, please.
yeah, I want to make 2 comments. So, again, in the cancer field, it's not necessary, but the FDA has now approved 2 genomic tests, Foundation Medicine and MSK Impact. So I think that's been, and they've actually received quite a bit of criticism for that, interestingly enough. But I actually can see the day, I would give it 5 years, where it will be malpractice not to do these tests in cancer. So I think that's very important. The other thing about cost effectiveness, I think this was a really important comment. So there's costs and there's cost per life year. The costs of these therapies are much higher than not giving them because, it's cheap to die. But it's the cost per life year that is really important. And I'm surprised at how many times I don't see that differentiation.
Yes.
Edmund Caparelli, pediatric clinical pharmacologist at University of California, San Diego. I just want to highlight some of the comments that Dr. Relling brought up about needing a great or a platform that we can actually work off that integrates the data. But I also want to remind people who went to the marvelous talk yesterday that showed that it's more than just the renal function, the hepatic function, the drug interactions, and the pharmacogenomics we need to work about. We need to think more broadly in terms of what are the alternatives, what are the consequences. And I think the example of efavirenz, but Dr. Telenti brought up just at the end here, discontinuing a drug, depending on the time frame, depending on the age, makes a big difference. We have a lot of alternative therapies, but people who discontinue because of side effects typically become non-adherent to those drugs. And so for an antiviral drug, you start taking intermittently, you build up resistance not only to the efavirenz, but to other drugs in that class. To complicate it even more, let's go down to kids. So I was involved in a study, Impact P1070, where we were doing pharmacogenomic testing after the fact, kids less than 3 years of age. What ended up happening is we had to close the study, revise it, because we couldn't do a post hoc analysis. We could not get the dose right without the pharmacogenomic information up front. So we actually developed a dosing guideline based on just one of the steps, not a full activity score, where we gave 1/4 of the dose in patients who identified as poor metabolizers versus those that actually had either intermediate or full extensive metabolizer status. The issue here is not just that we don't have those same alternatives in kids that we have to consider, but also all of the side effects that would cause one to discontinue the drug in terms of sleep disturbances, et cetera, are really tough to detect in preverbal kids. There was some recent information at IAS that suggests that maybe long-term early exposure to these drugs may have cognitive function long-term, even in patients who aren't HIV infected. So, this is really why the importance of having these implementation guidelines with the expertise is absolutely critical for not only just getting some platform integration, but also making certain that people have that broader perspective of alternatives and, what are the real consequences, not just in your adult populations, but, across the board.
Thank you. Yes, please. I think you were next. Yeah.
Hi, Deanne Pillars, University of Illinois at Chicago. I'm a neonatologist and want to add another level of complexity here, which was touched on a little bit with the acetaminophen. But we have a developmental overlay in everything that we do. We deal with drugs that have never been tested in babies, and those drugs may have been around for 50 years. But our patient population is considered ethically off the table to be doing drug studies in, so we really deal in a world with no answers. All of that said, I wanted to share with you on the education front a new initiative that's being kicked off this year within the neonatal community that's based out of the Vermont-Oxford Network, which is an international collaboration of over 900 neonatal intensive care unit groups, and the Rady Genome Institute right here in San Diego, to educate the neonatal community about genomic medicine through a combination of webinars and workshops with the intent of bringing everyone in our clinical community up to speed on these issues. And I think that kind of effort is going to need to be replicated across the board within medicine because, at least from my perspective, I think the medical community is even less prepared to deal with this new revolution than 30, 40 years ago because of the de-emphasis on science in medical education. So even new graduates from programs don't have a clue about what we're talking about here today. So I just want to go back to that very fundamental step of the importance of education, because I don't think the medical community is ready to embrace this if they don't understand what it is.
So a few very interesting points in your comments. Reactions? Yes. Razelle.
Yeah, so I have a reaction to your educational comment. I think it's really important. So we see our fellows. These are people that have gone through 3 years of college and 4 years of medical school and about 5 years of fellowship training, and they're coming out with, I would say it's pretty close to zero genomic background. When we bring them on as faculty, we have to begin to teach them as if they're like first-year college students. That really has to change. They have to come out as native speakers. They should be native speakers. They're young. They should be immersed in this right from the beginning. And not only are they not native speakers, but they've never even heard of the language. So I think your comment is really important.
George?
Yeah, one easy way to address this would be perhaps make it obligatory as part of your CME credits needs to be accumulated per year. So if we considered one, let's say, week-long course on genomic medicine, that it would provide you with enough CME credits, and this will be, of course, imposed by let's say, the regulatory bodies of a certain country, as it is being done at the moment in the UAE, for example. This would be one solution for that.
Amalio, did you have a comment?
It's a bit of I was thinking about education and what's the material we educate on, and I think they're all the tremendous efforts. But I wanted to tell another anecdote. I took 1 year 4 students from the pharmacy school, and I gave them as task to classify literature on transporters, genetics, with just very basic concepts. How many individuals were checked, how many variants, did they use multiple testing correction, the sanity of the statistics. And unfortunately, they pulled out 1,000 papers, and they came up with a list of 56 that were considered valid by the measurement of just very basic statistics. So I think we have an issue not only on educating the individuals, it's on educating ourselves about how to correctly analyze a score, do statistics on genetics, and understand what it means to do candidate gene analysis versus broad spectrum. For me, that was a sobering experience to throw away probably 900 papers of wrong, and I'm sure you have a comment on that, wrong claims.
Gert-Jan? Peter, did you have?
Well, in my view, I think what Deanne Pillars indicates is actually that the problem or the solution should start much earlier, and that's just get the kids' interest, but get more of this stuff on the curriculum. And what happens at curriculum nowadays are still like silos that are organized and disease organized, and so it's really difficult in this siloed situation to actually get some horizontal stuff like the genetics and the switch from genetic to genomics in the curriculum. But somehow this has to be done. And then after that, it can go as well in neonatology. And I would have thoughts on how to do at least some prediction, not now, but perhaps 10 years from now. And that's based on the genotype, phenotype in drug reaction from the genetic background of the parents. You could actually try to begin bootstrapping your way in how you would, or at least would consider correcting that for a neonate, rather than doing trials in neonates, because, well, that's the big difficulty, of course. But the thing is, this whole genetics, genomic stuff is really hard to find in most of the curricula. That's the same in Europe.
Yes. Peter, did you have a comment?
Yeah, I was just going to say, something I'm a big believer in is, after the formal, I think formal education is great, but, once people move on, either, as a practicing clinician or as a researcher, to sort of make opportunities for people from all sorts of different backgrounds to come together. So we do that, within the lab. If there's a clinician that's interested in the microbiome, I don't want them to stay in the clinic and just talk to us by Skype, but have them in bed within the lab so that they're shoulder to shoulder with people that are, chemists or biologists or come from these different backgrounds. And then I think that provides a training environment where they can really learn a lot more than they could from a course. And so, at UCSF, we're trying to do that more systematically, so it's not just lab by lab, but to have sort of spaces where people from all sorts of different backgrounds that share a common interest can come together and work together, in a shared environment.
Thank you. Yes.
Bill Loting from University of Minnesota. With next-gen sequencing, we're picking up more and more rare variants. And this is especially true in cancer, and now may even be important in the microbiome as well, as we find variation that affects function. And as we try to automate this, as that one woman was talking about, when we find these new variants, we're going to need to have a well-curated database where people can go to. I mean, there's a lot of databases out there with varying levels of curation. Some deal with functionality and some don't. But I think it's going to require a major database, which was a go-to database with these variants as they get identified, to try to determine, maybe through in silico ways at least, to try to determine functionality. And I'm wondering, is there a role for PGRN to deal with these rare variants, put them into a database, and then as more and more information is identified, we can start ascribing functionality to some of these variants. So as we go through these bioinformatic approaches, as we sequence an individual, we can start pulling out those variants that are functional and important, and hopefully ignore the plethora of variants that we're finding that we don't need to worry about.
Thanks, Mary.
There are tools relatively recently established as PharmVar, which Andrea Geidick is in charge of. She was in the room earlier. And that's a collaboration with PharmGKB. So there's a high coordination between PharmVar, PharmGKB, and CPIC for pharmacovariants, including rare variants, to try to have a common approach to assigning function. And ClinVar, which is with whom we also have a relationship, is doing the same for disease-causing variants. And they have
LOVD as well.
Pardon me?
LOVD as well. I mean, there's just a number of these databases that are out there.
You're right. There are a number, and we've got to coordinate so we don't waste our precious resources on reinventing the wheel, I think. But I think for pharmacovariants, we aren't reinventing the wheel. We're working together. It will eventually be a harmonized system. And as somebody asked yesterday, there's going to be some controversies about how to assign function to these variants, but you're absolutely right. It's got to come together in common systems that are used preferably worldwide. But I do think that we're on the way to having just a couple systems for that.
One more statement, and then George, I think, is good. But it'd be nice to have an invitation for clinical diagnostic labs to also feed into it as well.
They do.
But do they know about it? I mean, it seems like
they certainly know about ClinVar.
Yeah. Okay.
I think actually Amalio was first, George. Then sorry.
I think you brought, Bill, two important issues here. For maintaining a database, you need two aspects. One is funding. And I've been involved in database curation and development for over 10 years. And then I know that funding for this particular effort is very limited. So you have to use resources coming from other grants. And it can be, very painful in this respect. The other important aspect is motivation for data sharing. So unless you share your own data, you find in your own genomics expedition, and you keep it on your own, then it doesn't help the community. So that's why I think initiatives like PharmVar and the ClinVar, it's quite important so that they can motivate fellow researchers to share the data with the community, because at the end of the day, their findings will be important for the patients.
Thank you. Amalio?
Yeah, so I think more largely for genomics, to answer to your question, so there's today 800 million variants described across all the genome projects. So as you said, it's a huge issue of how we're going to annotate. So what Scripps research is providing, and it's open to people who want to contact us as a pilot, is something called Omni. Omni is just, it runs around and captures every single thing ever said about every single position in the genome, whether it's from ClinVar, from its frequencies, from whether it's an enhancer, whether it has a functional readout of any sort, and it's on a first pilot, and I'm happy to give, because it's a community project. It will be fully endowed for crowdsourcing. What you are asking, how can you just write something about it, and it will be fully equipped for intelligence. That means it will learn as people use it on what is being said. But of course, it's a completely different concept to a standard database. It's just tell me anything that has ever been said about that place in the genome.
Thank you. Yes.
Thank you. This has been fascinating. One of the panelists mentioned renal function as a potential stratification for the patients. We've had expert systems that will provide prescribing guidance on renal function for more than 20 years now, and in general, physicians don't like to use them. So I do a certain amount, so I'm a PhD, but I do continuing medical education in genomics, and if you are proposing to add the level of expertise to the existing fellowship trajectories for subspecialists, the system is just going to buckle. There is not enough years in people's lives to develop that level of expertise in the number of personnel that we would need, and they're not going to rely on these black box systems, nor, frankly, should they. I think that the only professional route in the system where there is really room to provide the kind of expertise that a physician could go to as a consult, which physicians will do, is in pharmacy. The pharmacists right now can be professionally certified just with their doctorate. There is room in that track to provide the necessary genomics expertise for people to have consultants. And I would like to invite the panel to disagree with all of that.
Great. Yes,
I have a comment. So I think that's a really important comment. I spent about 20 years at MD Anderson Cancer Center, and what MD Anderson does is it embeds PharmDs in the clinic with you. They sit right beside you. And it's, I think, a huge benefit for patients. They correct the doses for renal function, liver function, drug-drug interactions. They're very well trained in that area. They've done residencies in that area. I think it's a huge benefit for the physicians. I think, most importantly, it's a huge benefit for the patients. So I completely agree. The system of embedding PharmDs, like they're sitting right beside you in the clinic, is, I think, is huge. The other thing one can do, which hasn't become that popular even in research projects, but I think is really important, is therapeutic drug monitoring. And that's just looking at drug levels. So I know there's been several efforts to develop even bedside machines that will give you the drug levels. And it's sort of petered a little bit, but I actually think it's pretty important. We do it for a set number of drugs, some antibiotics and a few chemotherapy agents, but there's really no reason it can't be broadly applicable. Just let us know what, patient comes in, gets a little stick, what's your drug level, what's the normal drug levels, and let's adjust the dose. I wish I would see more in that realm.
I would agree with Razelle's points. I think we've worked for many years with PharmDs on most of our inpatient services at Brigham and Women's Hospital, and they've been invaluable partners. And not all of them, but some of them now have a fair amount of genomics, not practical genomics knowledge, which is also quite helpful. I think to the point of educating physicians in the genomics field, I wouldn't give up. In fact, I think there needs to be certainly more than the very simple genetics that most physicians learn both in the US and in Europe at this point in time. And it can be practically based, it can be basically elaborated in the context of cases, which is how many medical students these days in the US learn about complex disease. I think there are ways to at least get them facile with the terminology and with the potential applicability of modern genomics, recognizing that it's a very rapidly moving target and experts will be needed for consultation. Yes.
So I'm Andrew Monti, and I would like to step back here again and just actually reiterate that point. People's capacity for learning and knowledge is actually insatiable. And so I think that the point of steering away from any particular group is actually naive. We actually need to provide more education and more resources across the scope of education. And so the need for clinical advocates is actually really important. And each person in this room is a potential clinical advocate. Everybody has a different perspective to offer, and we teach different groups of people, and we teach a broad number of different people. And so actually, I would ask that each person in this room take that as a guide, right? We need to actually continue to educate every group that we come into contact with, and each one of us can do these small things. So yes, it's adding that little bit of pharmacogenomic education to your lectures. Yes, it's actually writing commentaries when the CPIC guidelines come out to your own individual journals and groups. It's offering to actually give these talks at societies. Each one of these small actions actually moves the bar that little bit. And so it's on all of us. We all have to do it.
Thank you. Yes.
Yeah, I wanted to follow up. It was Wolfgang Sadeh from Ohio State University. I wanted to follow up what the podium chair already mentioned, that there has to be more emphasis on gene network analysis or interacting networks that are dynamic. So far at the symposium, we have really seen a very strong emphasis on rare variants that have high impact. And then in complex phenotypes, we have polygenic scores that are also nicely predictive, but they're not, functionality is really not important. And then the emphasis on the large databases where how do you interpret single variants and so on, and the CPIC would be a single variant mostly, and a drug, and how to interpret that. And there's so many factors in there that we do not then consider. So I would just like to suggest that we do pay a lot more attention to the network in which these things are operating. I mean, nature has evolved. There's a lot of natural evolution that is not based on single variants, it's based on the interactions of multiple variants. And I believe that there's a very large dark room there that needs to be entered still.
Thank you for that comment. Actually, we just had the first International Conference of Network Medicine in Rome last month, and we're really expanding. It's beginning to catch on as a strategy. It certainly has in certain areas in smaller ways, but I think only in the last 5 to 7 years has it begun to develop some momentum. So I expect it will be in the pharmacogenomics world a little more quickly than perhaps you might have thought. Yes.
Well, I actually think that humans are networks of networks.
Yes.
And that means that going in from, say, predictive or prognostic, highly complex risk scores, which is now, well, doable but difficult, would be a fair way, because fundamentally you then will not have mechanistic insights, and it may be that you're looking at a very complex cloud of events. But then if you get something out of that, then you can probably parse this into groups that are similar, and then ultimately get groups where even clinical trials may become doable or direct prognostics. So I would fully agree that going for the rare variants with the big effects is, well, not very useful.
Right.
But the rare variants that together whisper a reasonably loud message is, I think, perfectly doable with the type of IT and bioinformatics that we have now, and the data are coming.
We have time for one last question. Yes.
Hi, I'm Grace Kuo from University of California, right here in San Diego, Skaggs School of Pharmacy and Pharmaceutical Sciences. So I wanted to respond to some of the education-related comments. In 2016 and onward, one of the accreditation criteria for all the schools of pharmacy in this country, at least, is to teach pharmacogenomics. The extent and the amount, the concept areas may differ from school to school, but in order to be accredited as a school of pharmacy in the US, we must teach pharmacogenomics. A few weeks ago, and so NHGRI has been instrumental in helping us do this. Several years ago, they helped us assembled a conference. It was by invitation only with about 10 different pharmacy organizations and residency training programs to help endorse this. And many years later, now we have finally made that into an accreditation requirement. And so recently, we also encouraged AAMC to make similar, if you so will, regulatory or educational requirements in all schools of medicine. Hopefully that will go somewhere, and we need everyone's help. The NIH has a group called G2C2. They have, so I have been involved with their work. It involves interprofessional organizations, including physicians, pharmacists, physician assistants, nurses, and genetic counselors with unique sets of competencies to teach genetics and genomics. And so we peer review all educational materials and post them online. So the NIH also posts and hosts the website. So if you're interested, please go there and submit any educational materials, whether it be website, whether it be articles or books that you would recommend. And so this group of editorial board will review them. In addition, NHGRI also has a ISCC group. It was not until recently when they now finally open it up so you as an individual or even industry can sign up to become a member. And so NHGRI has short course for educational purposes. Up to this point is for nurses and genetic counselors, and perhaps they're thinking about physicians or broadening it up to a wider scope. So finally, here at UCSD, we have pharmacogenomics.ucsd.edu with PharmGenet educational materials. We welcome anyone to collaborate to help update, but that information is free of charge, originally funded by the CDC, and it's open as a shared curriculum for all the colleges of pharmacy to use to teach pharmacogenomics.
Great. Thank you for your comment.
So can I just add to that?
Yes, Charles, go ahead.
I just want to say that NHGRI right now is in the midst of strategic planning, and there's going to be a meeting here to pick all of your brains in a sense in terms of where are we taking genomics, and I think pharmacogenomics is a big part of that discussion, and that strategic meeting is today at 7:30 p.m. In room 29. So again, if you have ideas about how to move these things, including setting up databases and who will guide this for the next 10 years or so, I think this is the time to speak up because it will help in terms of how budget and things are put in place.
Great. Well, thank you. I want to thank all the panelists and also the audience for their participation for a terrific session. Thanks.